Hello everyone. Welcome to the video lecture. I'm Dr. Rajati K from Biochemistry Department. In this video lecture, we will see about the topic gene bank. In this video, we will learn about the introduction, gene bank submissions, access to gene bank file formats, database record of gene bank entry. Introduction to Gene Bank. It is the best known nucleotide sequence database. It is available at the US National Center for Biotechnology Information, that is NCBI. We know that it is a public sequence database which in its present version contains over 12 trillion nucleotide bases in more than 2 billion sequences. This record is as of February 2021. It is part of the International Nucleotide Sequence Database collaboration, which comprises of DNA Databank of Japan DDBJ, European Nucleotide Archive ENA, and Gene Bank at NCBI. These three organizations exchange data on a daily basis. It contains DNA sequence of each and every organism. It gives data about every type of DNA from mRNA to cDNA. It is retrievable through NTRES. It gives information about ESTS, expressed sequence tags, and their protein translations, genome sequencing data, and sequencing polymorphism. It has a flat file structure, that is an ASCII text file, American Standard Code for Information Interchange, that is readable both by humans as well as the computers. Gene Bank, the data has been submitted by many processes using different softwares. It is built by direct submissions from individual laboratories as well as from bulk submissions from large-scale sequencing centers. Here, only original sequences can be submitted. Sequences can be entered into Gene Bank by anyone via a web page using Bankit or by email sequence. This in case of working with large sequence sets. Upon receipt of a sequence submission, the Gene Bank staff, what they will do now, they will examine the originality of the data and assigns an accession number to the sequence and performs quality assurance checks. The submissions are then released to the public databases, where each and everyone can retrieve the data by interest, or it can be downloadable by FTP, which is File Transfer Protocol. Bulk submissions of expressed sequence tag EST, then sequence tagged sites STS, genome survey sequence GSS, and high throughput genome sequence HTGS data are most often submitted by large-scale sequencing centers. It contains information like each and every data in the gene bank format consists of accession numbers, then gene names, phylogenetic classification, and references to published literature. This is the URL for the gene bank. Each data in the gene bank consists of the informations like sequence, accession numbers, the scientific and gene names. How the data can be accessed to gene bank? There are several ways to search and retrieve the data from gene bank. We can search gene bank for sequence identifiers and annotations with interest nucleotide. We can search and align gene bank sequences to a query sequence using BLAST, which is a basic local alignment search tool where the two sequences can be aligned. Then search, link and download sequences programmatically using NCBI e-utilities. Then ASN.1 and flat file formats are available at NCBI's anonymous FTP servers. So from these processes, we can access the gene bank data. So there are many file formats. The data are present in different file formats. We will see one by one. The first field command in GeneBank output the data is locus. So the locus field contains a number of data elements including locus name, sequence length, molecule type, gene bank division and a modification date. So which has been described below one by one. The first one in the locus field is locus name. So locus name will be like that SCU49845. So like that some 8 letters or 10 letters will be present as the locus name. The locus name, it was originally designed to help the group entries with similar sequences. So the first 3 characters here, for example, this SCU, what it represents now, it represents the organism's name. It will be designated there. The fourth and fifth characters were used to show other group designations, such as it may represent the gene product or the segmented entries. The last character was one of a series of sequential integers. Then the next part in the locus field is sequence length. So the sequence length, what it represents now, it represents how much number of nucleotide base pairs are present in the sequence record. For example, if there are 5025 base pairs, it will be represented as 5025 BP. So the length of the sequences will be represented. The next one is molecule type. The type of molecule that was sequenced, that is the output. It can include genomic DNA or a genomic RNA, then precursor RNA, even mRNA or the complementary DNA, then ribosomal RNA, transfer RNA, small nuclear RNA, and small cytoplasmic RNA. Then the next one that is present in the locus field is gene bank divisions. So the gene bank division to which a record belongs is indicated with the 3-letter abbreviations. So there are totally 18 divisions in gene bank database. So the first one is PRI, which represents primate sequence. ROD, it is a rodent sequence. MAM represents mammalian sequence. BRT for vertebrate sequences. INV for invertebrate sequence. PLN for plant, fungal and algal sequences. BCT for bacterial sequence. VRL for viral sequences. So PHG for bacteriophage sequences, SY1, SYN synthetic sequences, UNA unannotated sequences, EST expressed sequence tag sequences, PAT pattern sequences, STS sequences, GSS sequence, that is genome survey sequences, HTG high throughput genomic sequences, HTC unfinished high throughput cDNA sequencing, ENV for environmental sampling. Like that, totally there are 18 divisions in gene bank file format in that database. Okay. So the last portion in the locus field is modification date. So this is the last one. The date of the last modification was represented in the locus field. For example, if the data has been modified, that is by last month, that date will be mentioned there. That is called as the modification date. The next field command in gene bank file format is definition. So the definition, what it does know, it includes the information such as source organism, gene name or a protein name, or some other descriptions of the sequence's function. Even the non-coding sequences can also be represented. If the sequence has a coding regions, that is CDS, description may be followed by a completeness qualifier such as complete CDs. So like that it will be represented whether it is a complete coding sequences or not, or a non-coding sequence. Everything will be clearly defined in the file format. The next field comment is accession number. This is very important since it is found to be a unique identification tag that is called as an accession number. For example, see here, it will be represented as U12345. So this is found to be, this AN is accession number. This is found to be the permanent record which cannot be unchanged even if changes are subsequently made to the database record. In some cases, a new AN can be assigned to an existing number. For example, an author adds a new database record that combines the existing sequences. So in that case, a new AN can be added. Even then, the old AN is retained as a secondary number. Even the old AN will be represented as the secondary number. Okay. AN is the only way to absolutely verify the identify of the sequence or the database entry. So through this accession number, we can retrieve all the data. So we can compare it. So if anyone, again, if it is represented, means some modifications have been done in it. Then versions. The next field comment is version. So your nucleotide sequence identification number that represents a single specific sequence in the gene bank database. This identification number uses the accession dot version format implemented by GeneBank ENA DDBJ in February 1999. For example, see here U12345, that is the accession number for one file. So if a new version has been added, means they will represented as .1. If again some more version, means like the .2. So it will be represented, the file format, the version number will be included in the file format. Then the next one is keyword. Keywords which shows the words or the phrase which describes the sequences. Then source, the organism name will be mentioned. Organism, the scientific name of the organisms like genus and species name will be mentioned, and even its phylogenetic lineage will also be shown. As such on the phylogenetic classification scheme that is used in the NCBI taxonomy database. The reference of the data will be given. It is a citation of publications by sequence authors that supports the information presented in the sequence record. Several references may be included in one record. References, they are sorted automatically from oldest to newest. Various classes of publications can be present in the references field, including journal, article, book chapter, book thesis, even monographs, proceedings chapters, proceedings from a meeting, and a patent. Then the next field comment is GI, Gene Information Identifier. The sequence identification number has been given. Then the translation, the amino acid translation corresponding to the nucleotide coding sequence will be represented. Then gene, we know that it is a region of the biological interest identified as a gene and for which a name has been assigned. Then complement indicates that the feature is located on the complementary strand. So the next field comment is protein ID. A protein sequence identification number similar to the version number of the nucleotide sequence will be given. So protein IDs generally consist of 3 letters followed by 5 digits, a dot and a version number. So like this it will be represented. So AAA 98665.1. So again, if anything, the version number can also be increased. So that will be represented as 0.2. So if any changes are made, it will be Again, it can be modified as pointry. Like that, the versions will be increased. Then the next field command is base count. It gives the total number of adenine residues, cytosine residues, thymine and guanine bases in the sequence. So the total count will be represented. Then origin, this is the last one. It contains the sequence data which begins on the line immediately below the field title. This is one of the example file format that is present in GenBank databases. See here the locus. One example is given SCU 49845. That is the locus. Then 5028, the base pair, that is the length of the sequence. Then it is found to be a DNA sequence, that is the molecule is DNA. So PLN, it denotes the plant species. Okay. Then this data was published on 21st June 1999. So the definition of the data, that is it is a Saccharomyces cerevisiae. Then accession number is present. Then version, that is again the data version number, that is 0.1. Then GI number is given. If sometimes the keywords may be given or may not be given. Then the source of the data is from Saccharomyces cerevisiae, that is Baker's yeast. Then the organism name is mentioned there, whether it is an eukaryote or fungi or ascomycotas or like that. Everything, the subdivision, the phylogenetic lineage will be given there. Then reference, for example, the author name, the title of the journal where it has been, the data has been retrieved, the journal name, and the PubMed literature number. Even if more references has been included, that reference too will also be mentioned there. Then authors, then features, so from which source, then coding sequences, CDS, and the gene information, and then origin. So this is the final step. So this is the file format for the GenBank database. Okay. So I referred this data have been referred by following these references. So when we go with the summary of this topic, so we have learned about what is introduction, how the data have been accessed in GenBank, how to submit data in GenBank, then what are all the field commands that is present in the GenBank database. Thank you.